So let's talk about gram stains. So if the first one that we talked about, a simple stain, uses one type of dye, you don't get any sort of important information. Differential type stains, this is once again a positive type stain, a differential stain uses more than one type of liquid. So you can see here, this is what the steps of a gram stain are. There are four different chemicals that are involved in a gram stain. That's why it's called differential. It allows the gram-positive bacteria to come out a different color than the gram-negative bacteria. Now, if you refer back to the differences between our gram-positive cell wall and our gram-negative cell wall, the cell envelope, we have a large, thick peptidoglycan layer in one cell membrane in our gram-positives, a very small peptidoglycan layer, and an outer and an inner membrane in our gram-negatives. So the differences between their cell envelopes are going to allow the chemicals that you use in a gram stain to penetrate the cells in different ways. The ways that they penetrate the cells are going to result in different colors if you do the stain properly. Gram-positive cells will become purple, whereas gram-negative cells will become pink. Oh, I forgot to mention, these YouTube links are links showing you how to do a simple stain or how it works, talking through it, and sometimes a little bit of both. So you can see here our gram-positive cells by themselves, they are purple. Our gram-negative cells, they are pink. They are not red. And this is a microbiology rule. I did not make it. It is made by the National Association of Microbiologists. This is pink. So this is a mixed culture. You can see gram-positive caucuses and gram-negative rods. And this is the differential concept here. If you used a simple stain, everything in here would be the same color. You would know that you had a gram-positive species and a gram-negative species causing the infection in your patient. You would just see, oh, it's all purple, it's all gram-positive, it's all blue, it's all the same, and you wouldn't know that you had two different species causing the problem, and you might not give the patient the proper treatment. So differential type stains allow you to tell apart different species of bacteria and start the diagnostic process. So let's talk about how a gram stain works. So this is our little breakdown of our gram-negative cell wall over here. You see the outer membrane, our very thin little peptidoglycan layer, and our other inner membrane. Our gram-positive over here, it's very thick outer peptidoglycan layer, and it's one inner membrane. So I've put the gram stain solutions in order up here. Crystal violet, iodine, decolorizer, or alcohol, and safranin. And these are the four chemicals that are involved in a gram stain. So how does this work? Well, what we're going to do is we're going to pretend that you've got a mixed sample of a gram-negative bacteria and a gram-positive bacteria on your slide. You've heat-fixed them, and now you're going to start staining your gram stain. So the first thing that you're going to do is you're going to add some crystal violet to your heat-fixed smear. That crystal violet is liquid, and it's bright purple. It's going to penetrate down through the cell membrane of our gram-negative cell and penetrate down through the cell wall of our gram-positive cell over here. Crystal violet bonds to peptidoglycan. So this crystal violet molecule is going to embed itself inside of the peptidoglycan layer of these bacteria. You can see here, over here, our gram-positive has quite a lot of crystal violet embedded inside of its peptidoglycan layer. A very large peptidoglycan layer, it can hold quite a lot of crystal violet. Whereas over here, our gram-negative has a very thin peptidoglycan layer. He can't really hold much crystal violet, but he will take crystal violet in regardless. So over here in our gram-positive, you add some crystal violet in the first step. That crystal violet's going to bond inside of the peptidoglycan layer, a lot in our gram-positive, do that big thick wall, and just a little bit in our gram-negative cell wall because it has a very thin peptidoglycan layer. You're going to let that sit for about a minute or so, and then you're going to wash it away with water. Wash all the crystal violet away. Then you're going to add some grams iodine. Now, grams iodine and crystal violet, when they come in contact with one another, stick to each other like a magnet. They bond. They form a rather large crystal violet iodine complex, a big giant molecule called a mordant, which is just a chemistry word for big large molecule. So you pour the iodine on your slide, the gram-negatives and the gram-positives at the same time. The iodine is going to penetrate down through the gram-negative cell membrane, through the cell wall. It's going to come in contact over here, and the iodine under gram-positive, it's going to bond to all of the peptidoglycan, all the crystal violet bond in the peptidoglycan over here. So tons of mordant, tons of big giant globs of crystal violet and iodine in our gram-positive cell membrane, cell wall. Whereas over here, In our gram-negative, you have just a little bit of crystal violet and just a little bit of iodine that's going to bond to that. So a lot of purple bond over here in our gram-positive and just a little bit of purple that's bound over here in our gram-negative. So you're going to add the iodine and you're going to let that sit for about a minute or so. Then you're going to wash away the iodine with some water. Now at this point, gram-positive cells and gram-negative cells are both purple. They have crystal violet with iodine permanently bound inside of their cell walls. Over here in the gram-positive, quite a lot, and over here in our gram-negative, not a lot. The next step you're going to do is a very technique-based step. These are all just put it on and wait. The alcohol step or the decolorizer step is technique. Not enough or too long, and it's going to mess up your test. So this is all about technique and feel here. So you're going to pour some alcohol over your slide, or you're going to drip it. There are lots of different ways to do this, and you're going to let it sit for about 10 to 15 seconds. You're going to watch the purple run out of the cell walls. Alcohol strips away cell membranes. So alcohol breaks apart lipid bilayers. Now over here, our gram-positive cell does not have a cell membrane to rip away on the outside. Our gram-negative cell does. So the alcohol is going to come in contact with both the gram-positive and the gram-negative at the same time, and it's going to rip away the outer cell membrane of our gram-negative, exposing the peptidoglycan layer. Now the second thing that's going to occur is alcohol dehydrates cells. So alcohol is going to dehydrate and compress this peptidoglycan cell wall. It's going to suck the moisture out of it. It's going to dehydrate the cell. And when it does, it compresses this peptidoglycan cell wall membrane layer together. Which, when it does, it compresses and sticks inside all that mordant crystal violet iodine complex and crushes it all together inside of the cell wall, holding it all in there permanently. So lots of purple has been crushed and crowned together inside of the cell membrane, our cell wall, of our gram-positive bacteria. Now over here in our gram-negative, we've washed away the outer membrane. The alcohol did that. And now the alcohol is starting to wash away and dehydrate the peptidoglycan. It's starting to suck the moisture out of it. Well, these guys don't have much of a cell wall to begin with. And when the moisture inside of their cell wall starts to disintegrate, or starts to be sucked away, starts to dehydrate, their cell wall will break. And when it breaks, alcohol is able to wash away the crystal violet and the iodine complex from inside of that cell wall. It washes it away. So at the end of the alcohol step, about 15 seconds or so, you are left with a compressed gram-positive cell wall with lots of permanently embedded crystal violet and iodine inside of it. This gram-positive cell, is at this point purple. It will remain purple for the rest of the test. Prior to the alcohol step, our gram-negative cell was also purple. You added the alcohol. It stripped away the outer membrane of our gram-negative, dehydrated the peptidoglycan, ripped out the crystal violet and iodine that was bound inside of the peptidoglycan layer, and now you're just left with the outer membrane of that gram-negative bacteria. So there's no color the inner membrane. There's no color left on this gram-negative cell. So you just have an inner membrane with no color on it. So this gram-negative cell is colorless at this point. The next thing you're going to do is wash away the alcohol very quickly and add your safranin. Safranin's blood red. It gives it a very, very, very nice pink color underneath your microscope, but it's blood red on person. And you're going to add the safranin to the slide. Now, safranin's going to penetrate both the cells. You add a little bit of pink on purple, it does not matter. This thing's going to still be purple. There's not a lot of places in here for the pink to bound. It's mostly going to be crystal violet. So what little tiny pink does bother to bind to the cell wall does not matter. Its cell is going to still be purple. This guy has no cell membrane at all in the beginning to keep stuff out, no cell wall to have crystal violet bound into, so no color in him at all. All of his color has been removed. So you add some safranin to him. He's colorless. Safranin's pink. Safranin's going to go right through that cell membrane inside of the cell and give it a nice, beautiful pink color from the inside. You wash away the safranin after about a minute or so, and you're done. You are left with permanently purple gram-positive cells and permanently pink gram-negative cells if you do this effectively, if you do it correctly. Now, I mentioned the decolorizer alcohol step was technique-based. If you leave the alcohol on the cells too long, it will compress the gram-positive cells and break apart their cell wall as well, which strips all the purple out of them, which can cause them to stain pink with the safranin. So you can accidentally cause the gram-positives to show up gram-negative if you're not careful. So that's where the technique comes into. So gram stains are the very first step of diagnosing patients. You get a patient sample, you grow it in a lab. The very first thing you're going to do is gram stain it. If it's gram positive, you'll go on this step over here for diagnostics. If it's gram negative, you go on this step over here. The antibiotics or biotics for each one of these do not work on the other one for the most part. So this is a very, very, very important step to be able to learn to do properly so you can start the diagnostic steps. Now, as I mentioned, you will see the gram-positive cells becoming purple, and our gram-negative cells are pink. So over here, you will see the walkthrough of how to do this step by step, the walkthrough of what's actually happening through here inside of the cell membrane here, inside of the cell walls, inside of the peptidoglycan for our gram-positives, and the same thing over here for our gram-negative. So that's kind of a cool little walkthrough of that. So gram stains are very, very cool. Once again, purple for gram positives, pink for gram negatives. So gram negatives, gram stains allow you to see size, shape, arrangement, plus gram reaction, which is the most important step to diagnosing your patients.